Hi guys, welcome to Lecture 1 in Modern Genetics. We studied classical genetics with Charles Darwin, and we've actually been looking at modern genetics quite a bit over the course of the year. Modern genetics is really molecular genetics, and so molecular genetics, of course, is the analysis of DNA as these factors of inheritance. We're going to look at a couple important aspects of DNA and its ramifications as we move towards the study of evolution. One of the things that can happen to DNA is that it can undergo changes, and so we're going to look at these things called mutations and what causes them. We'll be able to look at the origins of difference between different species, and then we're going to end it all with some look at some diseases, and then finally ways to manipulate DNA, maybe even medical treatments. So what is a mutation? A mutation is simply a change in the sequence of DNA within a gene. So you recollect that DNA is a double helical molecule, 3 billion base pairs long, 3 billion letters long, 3 billion nucleotides long, and of course, you've got 21,500 genes in the DNA. And if I change a letter by accident, I can change the amino acid sequence of proteins. Now, remember that most of the DNA in your body doesn't code for proteins. Only about 2% of the DNA is coding strands. So mutations are relatively rare. And we'll go into that in great detail. First, what causes mutations? Well, they can be spontaneous. So remember that every cell in your body's going to have a lifespan, and it's going to, at some point, undergo reproduction for the most part. And when it replicates, it's going to have to make a copy of its DNA. And there's simply mistakes made in that copying of the DNA. And so that's one source. And then another source is environmental factors called mutagens. Okay? So some powerful mutagens include X-rays and UV radiation, organic chemicals in cigarette smoke and pesticides. And so the question is, what's the most likely mutagen you're going to be exposed to today? Well, you can see from our table here, there's a lot of different kinds of butterflies. How did it happen? Did all these different butterflies suddenly appear? And the answer is no. The truth is that there's an important theory of evolution called descent with modification that says that we all descend from some other progenitor species, and things change over time. So there was an original butterfly or multiple butterflies, and they underwent reproduction, and they changed over time and given us this myriad library of different butterfly types. So the source of this change is process of mutation. And so mutants are individuals whose alleles have been changed. And of course, while you and I share an extremely high percentage of homology in our chromosomes, there's definitely going to be some differences. And those tiny differences in the DNA sequence between you and me, or you and your father or mother, or your brother or your sister, account for the differences you see in our phenotype. And of course, there's lots of differences at the molecular level you can't see, like changes in the keratin protein or changes in the insulin protein or the hemoglobin protein. So remember that alleles are different copies of the same gene, and that the origin of the differences between those alleles is mutation. So, are you a mutant or are you normal? Well, remember that we study transcription and translation. And so in the transcriptional process, we take DNA and we copy it in a messenger RNA. And this is where a mutation can occur. Or there could be simply a mutation in the DNA sequence originally. And remember that there's this box called the genetic code right down here, the genetic code. And each 3-letter codon codes for a specific amino acid. So imagine if I changed one of these letters, if I changed this U to maybe UAU, what would happen? Well, I would end up with tyrosine, here's UAU, instead of phenylalanine. So my protein sequence code would be changed, which probably would give rise, or potentially would give rise, to a change in that protein structure and a change in its function. Now, lots of times mutations don't have an effect. The next level is they have an effect, which is not good. And then last but not least, rarely, you have a mutation that changes a protein in a good way. And those kinds of mutations, those kinds of things lead to something called adaptation. So here's your table over here. Here's all the different codons that code for the different amino acids. There's 20 essential amino acids. Can you name any of them based on the 3-letter abbreviation here? Hopefully you can. Well, there's a couple different types of mutations. Point mutations and frame shift mutations. In a point mutation, you simply change a letter in the DNA from maybe an A to a T or to a G or a C. In a frame shift mutation, you either take a letter out or add a letter in, and that's going to have a big effect because that sort of changes the sequence all the way downstream. From that letter on, everything down the road is going to be screwed up, and I'll show you exactly why. If you remember, back here in the genetic code, there is a difference in where the mutation occurs. Which letter in the codon is most important? Is it the first letter, the second letter, or the third letter? Which one's going to have the biggest change? So, when we talk about point mutations, we have 3 types of point mutations. The silent mutation. I can change a letter in the codon, say from UAU to UAC, and I will still get the same amino acid. All right, so remember from our studies before that the genetic code is redundant. Both UAU and UAC both code for tyrosine. Or I could have a point mutation called a nonsense mutation. And in this case, we change a single nucleotide, and this will change the amino acid coded for from its original amino acid to a stop codon. And the stop codon tells the ribosome and the messenger RNA that they're done and they disassociate and they end up cutting off the protein. Last but not least is the missense mutation. And in the missense mutation, I have an actual change. So a single nucleotide change can end up with a change in amino acids. And of course, that changes the shape of the protein and it changes its function, usually for the worst. And an example of that is sickle cell anemia, which affects the protein called hemoglobin. So what does hemoglobin do? Well, hemoglobin is the protein in our red blood cells that carries oxygen. And we know its sequence. And here's a short segment of its amino acid sequence, the first 7 amino acids. Well, in the person who has sickle cell anemia disease, one of the codes has been, or one of the letters of the code has been changed. And instead of having glutamic acid, it has valine. And that change, that single amino acid change, changed the shape of the hemoglobin protein, which ends up changing the shape of these cells. Now, this disease is endemic. That is, it's relatively common to people that that are from sub-Saharan Africa, and of course, lots of African Americans have this same disease, which is a very painful disease. One of the interesting things about the sickle cell disease is that if you live in sub-Saharan Africa, where there's lots of malaria, it can actually help you out a little bit. We'll talk about that later. So, once again, here are the different types of mutations, point mutations. If it's silent, I can change UUU to UUC, and I still get phenylalanine. Okay, so that's a silent mutation. I make a change in the genetic code, but it doesn't affect amino acid sequence. Another possibility is the Missense mutation, where I change, say, CUU to CCU, and I go from making a coding for the amino acid leucine to coding for the amino acid proline, which is likely to have a change in the structure of the protein, like in sickle cell anemia. Of course, last but not least is the nonsense mutation. And in a nonsense mutation, I take something like the codon UAC, change this letter to UAA, and that makes a stop codon. So the process of translation will just stop. Here's some examples with the RNA sequence and the amino acid sequence. I can go from GCA here, which codes to alanine, to GCU, and still have alanine. Silent mutation. I can have a missense mutation, where GCA codes for alanine is changed to GUA, which codes for the amino acid valine, like we saw in that genetic disease, sickle cell anemia. Last but not least, I can have a change here, like, for instance, going from UAC to UAA. Instead of coding for tyrosine, I code for a stop codon here, and then nothing else is translated downstream. So those are the point mutations: silent, missense, nonsense. One of the ways I get kids to remember the silent seems like it's a pretty easy one to remember, like nothing happens, you can't hear it. But I always had a tough time remembering the difference between nonsense and missense. Let's stop your nonsense. Get it? If you have a nonsense codon, you get a stop codon. Which brings us to frame shift mutations. Now, the frame shift mutations have to do with the frame shift that we're talking about is the reading frame. So if you recollect, RNA is read in a three-letter sequence, three-letter words we call codons. If I add or delete a nucleotide, I can shift the whole way that transcript is read. Kind of tough to talk about, but let's look at this example. So here I've got the sentence: the fat cat and the red ran, all three-letter words. If I added a nucleotide, that is, if I added an N here, then, and I kept the rest of the letters, but if I'm still reading in three-letter words, I would move the reading frame downstream, and everything after this word would be wrong, at least in terms of the appropriate amino acid sequence. Or if I delete one, everything moves this way a little bit, and I still have got a wrong sequence. Here's an example with amino acids and nucleotides. So met arg-valotyrosine, UAC codes for tyrosine. If it's down here. If I deleted if I added a U here, but GUC, everything downstream is going to be wrong. Frame shift, the reading frame is shifted. Similarly, if I have a deletion mutant, that is, if I get rid of this C here, everything will be shifted this way, and everything downstream of this transcript will be wrong. These types of frame shift mutations cause non-functional proteins. And a classic example of a deletion mutation is cystic fibrosis. So cystic fibrosis, CF, is a deadly disease that makes for thick mucus, and it has to do with the elimination of these three letters. Well, we're going to look at genetic disease in detail.